Okay, we're going to move on to the study of phylogenetic trees now, and the study of phylogeny and how different organisms are related to each other. So, phylogeny is a representation of the process of evolution that we present with models called phylogenetic trees, shown here on the top right. And these phylogenetic trees show species at the tips, common ancestors at the nodes, and they also common descent from a single ancestor. Time is moving forward as you move to the right of a phylogenetic tree, or in this case moving up in the one I put on the top right. Organisms all derive from a common ancestor, and subsequent species accumulate differences post-divergence. Trees were used first in 1801 by Augustin Augier, where he produced a rudimentary phylogenetic tree. This was then adapted by Lamarck in 1809, and then Darwin produced his kind of rudimentary phylogenetic tree in 1837, before the first fossil tree in 1840. It's not until 1879, which is shown on the bottom right here, where humans were first placed within the phylogenetic tree, showing how we related to other organisms. So we can determine the relatedness of species and where they fit on the phylogenetic trees through a few different methods. The first one that we're going to talk about is the study of morphology. The production of phylogenetic trees has created a school of biology that helps us better understand evolutionary biology and genetics. Morphology is one commonly used method of determining ancestry. Determining relatedness based on shared derived characteristics. The morphology method identifies ancestral states as outgroups, and derived characteristics from such groups determine descent. The evolutionary explanation with the fewest number of evolutionary events is generally accepted, and we term this a parsimonious explanation. So the explanation that requires the fewest number of evolutionary events to explain. So like when we talked about the dragons, a dragon evolving wings from its forelimbs is far more likely than it first sprouting 2 extra limbs on its back and then adapting those 2 extra limbs into wings. The morphology method is limited by instances of homoplasy, where similar states evolve independently in different lineages, which can be caused by convergent evolution, parallel evolution, and reversal. So the example I put on the bottom right here of homoplasy, we can see 3 different types of wing produced by 3 very different groups: the birds, which are the avian dinosaurs, the pterosaurs, which are avian archosaurs, and bats, which are obviously avian mammals. And all these traits have evolved through convergent evolution, but obviously we know that these species are not closely related, they're very distantly related, but these instances of homoplasy to produce a wing, if we didn't look too carefully, you might be inclined to think that, okay, all these organisms have wing, they must belong to one group where they actually evolved these structures independently. One example that's really good if we're talking about using the morphology method to identify species relatedness, if you look at the images I put on the top here, you can see a manatee on the left and an elephant's legs on the right. If you look at the kind of kind of nails at the end of the manatee flipper and the nails at the bottom of the elephant's feet, they both have these similar looking kind of nails on them. Now that's because both manatees from the order Sirenia and elephants from the order Proboscidea. They're both members of a group called Panungulata, the almost ungulates, or uneven ungulates, some kind. The Panungulates. These kind of nail structures on the manatee flipper and the elephant feet are because their shared morphology is based on recent common ancestry, and then they've both been adapted to their various lifestyles. So the manatee flipper has been adapted to a flipper, and in elephants they've been adapted to the kind of like stocky legs that can hold up an elephant. We also have the genetic method, where genetic analysis can be used to determine species relatedness via analysis of the molecular clock, which measures the accumulations in mutations in genes in different species to calculate how long ago the species diverged by comparing them to the expected mutation rate. Gene trees can also be constructed, which determine the evolutionary history from analysis of a singular locus, typically from a mitochondrial nucleoid. These are easier as you don't have to sequence the entire genome. Gene trees, however, are limited, and they depend on the locus that you use, because of incomplete lineage sorting caused by hybridizations between species, where a locus may be introduced to one species from a different species, so it's not kind of I forget the words. It's not kind of like a monophyletic passing down of a gene. The gene for the locus that we're talking about is traveling between species and having it's kind of messing up our understanding of the tree from which these organisms evolve, because the allele is passing from one species to another species, and those species have they have different evolutionary histories, although similar, obviously, because they can interbreed. As well as admixture from the inbreeding between previously isolated populations. So a few definitions that we need to know about the phylogenetic trees. We have the nodes, we have the branches, we have the tips, which we talked about in the first slides, and branch lengths are substitutions for the passage of time. We need to know that clades are a monophyletic group most of the time. And with regards to clades, modern taxonomy sorts animals into clades, but this isn't always the case. For example, lizards are not a monophyletic group, as some of them are more closely related to snakes than others. So we try to sort lizards into their own clade, but we can't. They're not really a monophyletic group. Some lizards are more closely related to snakes than they are to other lizards. And then a monophyletic group refers to all the descendants of a single common ancestor which make up a clade. Some uses of phylogenetics. Phylogenetics allows us to understand the evolution of novel traits, unique traits to certain groups. Phylogenetics also gives us information on things like the dwarfing rate of organisms on islands. For example, the Mediterranean dwarf elephants that became gradually dwarfed from their large ancestor Paleoloxodon. Phylogenetics also permits us to determine evolutionary shifts in lineages to determine when specific traits emerge for what purpose. And phylogenetics also permits us to separate lineages into different species if the data compels us to do so.